Over the past few decades, researchers have made groundbreaking strides in cancer treatment. Imagine unlocking a hidden cure within our own bodies. That's the promise of cancer immunotherapy. From the late 1980s to today, intensive research efforts have illuminated the incredible potential of our immune system to combat cancer, much like it defends against viruses and other diseases. To grasp the impact, let's consider a real-world scenario. Picture a close friend or family member facing the challenges of cancer, and the hope that arises when we discover our immune system can be fortified to stand against this formidable foe. And while on its own, our immune system is not always powerful enough to target all types of cancers, immunotherapy provides the much-needed boost to get us there. Now, let's dive into the world of immunotherapy, where treatments fall into two main categories: passive and active. Think of these as two strategic arms in our defense against cancer. Passive therapies possess intrinsic anticancer properties, acting as vigilant sentinels against malignancy. Examples include tumor-targeting monoclonal antibodies, oncolytic viruses, and the groundbreaking chimeric antigen receptor, CR, T-cell therapy. Active therapies, on the other hand, only unleash their anticancer effects upon engaging our immune system. Think of them as activating a dormant superhero within. Examples here include immune checkpoint inhibitors, anti-cancer vaccines, and cytokine treatments like IL-2 or interferon gamma. The complexity of these therapies' mechanisms can be overwhelming, so before we delve into them, let's simplify the complex gears that drive their actions. Take T-cell transfer therapy, for instance. Picture this as boosting our immune warriors in a lab, enhancing their ability to identify and destroy cancer cells. It's like turning our immune system into a specialized force capable of recognizing and eradicating cancer threats. Millions of these enhanced T cells, which were taken directly from the patient, are then infused back into their body, ready to wage war against the cancer threat. You can also think of immune checkpoint inhibitors as keys unlocking brakes on our immune system, allowing it to go full force against cancer. These drugs block so-called immune checkpoints on T cells, like PD-1 and CTLA-4, preventing cancer cells from exploiting them to evade detection. To understand this, picture immune checkpoints as safety mechanisms. After T cells are activated, these checkpoints are upregulated to minimize unnecessary or excessive T cell activity. However, cancer can manipulate this inhibitory system, essentially preventing T cell activity before it can even begin. Checkpoint blockade is like removing hurdles and letting the immune system go all out in its fight against cancer, disrupting one of cancer's many immunosuppressive traits. These brief descriptions are just scratching the surface of more complex, nuanced cellular interactions that exist within these treatments. Nevertheless, the biggest takeaway here is that, despite their complexity, the main principle driving immunotherapies is their ability to fuel the immune system and invigorate a powerful anti-cancer response. With this in mind, what's the immunological context behind these anti-cancer responses? Think of cancer as a crafty imposter disguising itself as part of the body. Immunotherapy aims to expose this deception, kickstarting an immune response against these mutated versions of self. Generally speaking, the immune system is a complex network of organs, tissues, and cells, and the substances they make. Its main purpose is to rid the body of pathogens, such as bacteria and viruses. Over the years, we have learned that cancer cells are no different. They are foreign and usually sensed just the same as any other invader. Now, meet the main characters in our immune story, CD8 cytotoxic T cells. These are like the superheroes of white blood cells, well known for fighting off viral infections. They are activated when dendritic cells present protein fragments, or antigens, produced within your cells via a cell surface molecule known as major histocompatibility complex, MHC-1. Quick side note, there are two classes of MHC molecules, MHC class 1 and MHC class 2. While all nucleated cells in your body express MHC-1, MHC-2 is more restricted and found mostly only on antigen-presenting cells, APCs, like dendritic cells, macrophages, and B cells. MHC class 1 only activates CD8 T cells, while MHC2 only activates CD4 T cells. Antigen coming from inside the cell is primarily loaded on MHC1, while antigen produced outside the body, such as from an extracellular bacteria, is loaded on MHC2. However, DCs are able to partake in a process known as cross-presentation, whereby exogenous antigens can also be processed and loaded onto MHC1 to activate killer T cells. This is very useful, and in the case of cancer, required at times when tumor-associated antigens are derived from proteins that are not typically expressed endogenously. As such, DCs can take up antigens from dying tumor cells or other exogenous sources, process them, and cross-present them on MHC1 molecules to activate cytotoxic anti-tumor response. These specific T-cell DC interactions are facilitated inside your lymph nodes, which are bean-shaped like highly organized organs scattered throughout your body, where circulating lymph drains to and out from. DCs can be thought as the surveilling police forces of your body, constantly sensing and processing antigens in the periphery, maturing upon receiving the right set of danger signals, and eventually migrating to your lymph nodes, where naive populations of T cells also reside, waiting to be activated upon the right set of peptide MHC interactions. These physical interactions specifically consist of the peptide MHC complex on DCs and antigen-specific receptors on the surface of T cells, which are referred to as TCRs, binding together alongside a CD8 molecule that identifies the T cell as CD8 positive. Co-stimulatory signals are also required before a naive T cell that has been presented with its corresponding peptide MHC complex can become fully activated. So up to now, you have a police checking out a suspicious character, cancer. The antigen presented is like an ID badge. If it is suspicious enough, the police will take a copy of the ID, navigate to headquarters in the lymph node, and present it to the commanding T cell officers so that an arrest can be made. Co-stimulation is like getting a green light from the chief investigator. It's the second signal for our immune response to intensify the attack against suspicious characters. Signaling molecules, known as cytokines, then allow the activated T cells to proliferate, while chemical cues, known as chemokines, guide them from the lymph node to the cancer. A final interaction between the T cell and the peptide MHC complex on the cancer cell itself takes place, allowing for these killer T cells to release their toxic granules and kill just those foreign, mutated cells. Fast forward to the horizon of cancer immunotherapy, and the landscape is truly exciting. Not only can we treat existing tumors, but recent research hints at establishing a lasting immune response that safeguards against future cancer threats. From cancer vaccines to discovering more exclusive tumor-specific antigens, the possibilities are vast. So, as we conclude our first journey into the immune arsenal against cancer, remember that the answer lies within us, within our resilient and versatile immune system. If you found this intriguing, give us a thumbs up, subscribe for more immunology content, and share your thoughts in the comments. This has been the Immunology Chronicles, reporting on our immune arsenal against cancer. Join us for the next episode as we unravel more mysteries in the realm of immunotherapy.